Okay, good afternoon, everyone. I think we'll get started. I appreciate those of you that are here over your lunch break. I'm guessing maybe the party last night was great, so some people maybe don't even need lunch today. So our lunch and learn presentation today is really to present to you some of the exciting developments that have been taking place within molecular devices, around what we've been doing in innovation in the 3D biology workspace and developing some new techniques to help enable 3D biology workflows. So as you've probably already gathered, the presentation today is going to be a presentation of two halves. I'm joined today by my colleague Angeline who will be taking the second part of the presentation. But to begin with, I'm going to start the presentation just by telling you a little bit about the use case for organoids and some of the work that we've been doing to develop a bioprocess for the large-scale expansion of organoids. And then when I'm done, I'll hand over to Angeline, who'll be telling you a little bit more about the work that Molecular Devices has been doing to automate the organoid culture process and also to automate organoid-based drug screening. Okay, so I think really, everyone is at this conference because we know that better models are needed, and the main reason that better models are needed is because of the so-called Eroom's law. So Eroom's law was coined in around 2012 by Jack Scannell and colleagues in the UK, and what it really says is that despite the fact that we're increasing our knowledge of biology and our ability to discover new drugs, actually what's happening is that drug discovery is becoming less and less efficient over time. So really paradoxical to where you would expect the field to be going, considering the advances in technology that are undergoing at the moment. And so part of the initiative to combat this lack of effectiveness within drug discovery and this great attrition that's within drug discovery pipelines is the requirement for new models. And I think as we've heard multiple times today, 3D models are really starting to gain traction because they really straddle that line between the sort of utility and the complexity. So they provide additional complexity without sacrificing too much around utility. So in particular, we focused our 3D models on organoids in particular. And we've really focused on organoids because they're much more representative and predictive than 2D cell models. And again, their predictive nature has been shown in publications that are now actually 5 years old. And here's just an example of a seminal publication that came from the Institute of Cancer Research in London. Organoid models were much quicker and cheaper than animal models. So again, there's been publications that have come out that show that if you use more sophisticated 3D models, including organoids, you can shorten the drug development time, and that's compared to going into animal models earlier in the process. And organoids are obviously also able to model a much broader range of mechanisms than just 2D cell lines alone. So for example, if you're looking for compounds that impinge on stem cell behavior or impinge on something that's specific to 3D biology, so cell-cell interactions or anything that's structural, you really need to be using a model that is more complicated than 2D biology. And so, I think we've heard in multiple presentations throughout the course of this conference that people are starting to use 3D biology and starting to use organoids, but we're really still not seeing widespread adoption of organoid technology. And the reason for this really is that there are still barriers to adoption, and these barriers can take numerous different forms. So the types of things that we really hear at Molecular Devices is that organoid culture is too time-consuming, it's too laborious, the reagents are too expensive, the skill needed from operators is high, there's often a high amount of operator-to-operator variability because the culture process is so difficult, the cost is high, reproducibility is not as good as it would be in 2D, cultures can be heterogeneous, and then the downstream analysis of organoids is much more complicated and much more difficult to perform. And so really all of these barriers, actually Molecular Devices sees as opportunities because these are all problems that we can solve. And so this is kind of what we set out to do, and we've developed a tool set to help solve some of these issues. And so the first item of technology that I'm going to tell you about that Molecular Devices has developed is a bioreactor-based bioprocess for the large-scale expansion of organoids. So really what this enables is the production of organoids in large volumes, which much better reproducibility and consistency than could be obtained by manual culture. And again, this basically addresses a number of those issues that I raised a couple of slides ago around needing skilled operators to be able to handle organoids, culture organoids, and get the level of reproducibility required. So this just provides an overview of the bioreactor-based bioprocess that we've developed to culture organoids. And so really what we do is we go from an established organoid culture, so organoids frozen down in vials in small numbers. From those we can make tiered banked stocks, and from that we can then go into the bioreactor-based expansion process. What we get out of the end of the process is cryovials of organoids that are standardized, all frozen in a large batch, all at the same time. And the advantage really is that all of those organoids have been handled according to set SOPs, so that operator-to-operator variability is minimal. They've all been handled using the same reagents, the same batch of matrigel. So really we're trying to reduce all of those points of variability other than obviously what is innately variable within the model systems, because one thing that we're very keen to ensure is that we're not destroying the model system while we're going through this process. The other thing that we've really developed that sits behind this, which often gets forgotten but is really important, is a robust quality system. And so that really just ensures that everything is done to the same level of quality, all of our reagents are traceable, and then also means that we have a robust method for quality control of the end product to make sure that what we're producing is fit for purpose and is every vial in the batch is the same. In terms of the benefits of the organoids that we produced, I've just sort of highlighted a few of those as I've talked about the process, but really we can produce very large numbers of organoids in single batches. So at the moment we're capable of producing between 5 and 6 million organoids. This is kind of a ballpark figure. It can be much more or much less depending on the exact organoid line. We're sort of at the moment working on our second generation bioprocess, which will be able to generate probably 4 to 5 times that amount. So these are very large batches, which really enable organoids in high throughput screening applications. As I've already mentioned, when we're producing each vial, every vial comes out at exactly the same passage. All the reagents that used for each for the batch have been the same, and so what that really gives you is really nice standardization. So the idea is that you can take one vial or you can take 4 vials and use them all at the same time, depending on the exact scale of assay that you want to use. Another significant advantage of the organoids that we produce is that the organoids are cryopreserved, and they're cryopreserved in a format that's effectively ready to use. So there's no subculture that's required by operators, so this isn't the same as organoids that you would maybe purchase from a biobank that you then need to expand manually yourself. So the idea is that the organoids can be taken out of the freezer, thawed and plated, and then within 48 hours they're ready to go into an assay. And so the power of that really is that a scientist or an operator can sit in the lab on a Friday, design their experiment, thaw their cells, and on Monday go with the experiment straight away, apply their compounds and have their results within a week. So it's really taking out some of that sort of laboriousness, the time involvement, and again also some of the skill that's needed to be able to culture organoids from scratch for every single experiment and in the same way every time. So really what we're aiming for is to make organoids more of a consumable rather than something that you need to factor into your experimental workflows to grow and generate every time you want to make. So if this all sounds good to you, then actually the way that you could access this within Molecular Devices by using our custom expansion service. So we provide bespoke contract development and manufacturing of organoids. So if you're interested in that, definitely come to the booth while it's still here today and talk to me about it. And basically what we'll do is we'll work with you to develop a statement of work to produce your organoids to your specifications. So we will take your organoid line, we will QC it, onboard it, bring it into our process, expand it, produce a large batch of organoids and give those back to you basically in ready-to-use format. So then you can have the organoids in your own freezer. When you want to do your experiments, you just take that out and go. We can also customize the level of quality control that we produce. So if you want samples of every passage or every 2 passages so that you know what you're getting every time, we can work with you to sort of develop a bespoke QC package so that you're confident that what you're receiving is exactly the same as your organoid line, which we're obviously confident that that's the case as well. So I've just got a few slides left before I hand over to Angeline. And in those slides, I just really wanted to show you some data because one question we get asked all the time is like, oh, what is this crazy bioreactor and is it going to do something insane to my organoid line? Is it going to change the phenotype? Is it going to change the behavior? What do the organoids actually look like when they come out? So in the next few slides, I'm just going to take you through some data that compares bioreactor-grown organoids with manually expanded organoids. And the experimental outline for what this looked like was basically we took the same starting culture of organoids. We grew half of the organoids manually, so just according to standard set protocols that are available in the literature, all the standard lab protocols. The other half of the culture we expanded and put through our bioreactor-based expansion process. We froze them down as product vials, so exactly how we would. We then thawed them, let them recover for 48 hours, and then comparing the manual harvest to those that have been freeze-thawed and recovered, we then put them through our analysis package, and our analysis package included immunohistochemistry, whole exome sequencing, RNA sequencing, and the drug response assay. So the organoid line that we did for this experiment, so actually we tested 3 different breast cancer lines. I'm just presenting the data today of one of those lines. On the left is a bright field image of what the organoid looks like in culture, so you can see it's a really nice, nice healthy growing line. I think this was actually an image of one of the bioreactors in culture, so really dense, very happy growing. We looked at the characteristic marker profile of the breast cancer organoids. And again, comparing manual and bioprocess was no different. This particular line is progesterone receptor positive, ER negative and HER2 negative. It's a line that's deficient for p53 and expresses the markers cytokeratins 14 and 18, which basically shows it's a basal type of breast cancer. So again, looking at the immunohistochemical staining, we saw no significant differences either in the expression or the pattern of expression of any of these key markers, showing that there's no subtype change between manual or bioprocess culture of this line. We then went on to look at genomic level analysis of the same line. So again, comparing manually cultured organoids, which is all the stuff that's shown in black text, so the top and the bottom here, to organoids that were grown in the bioreactor. And basically what we found is that the underlying driver mutations of the line were unchanged, whether the line was cultured through manual process or through the bioprocess. So we weren't losing or accumulating or in any other way changing the genotype of the line. And again, looking at the RNA-seq analysis, which is arguably more sensitive, here we're just showing a subset panel of which is the PAM50 diagnostic panel for breast cancer. And basically we're seeing very little changes. And again, this was true across the board for all of the breast cancer lines we tested and also for all of the RNA targets that are outside of this particular panel. This is just for example purposes. So as I say, really seeing very minimal changes between manual and bioreactor culture, at least over and above the sort of small changes that you would see between individual replicates anyway. And then finally, we put our bioreactor-grown organoids into drug assays. And again, we tested a panel of standard of care therapeutics for breast cancer. And really what we found, as we would expect, is that the bioreactor-grown organoids showed exactly the same drug response profile as the manually grown counterparts, again, just reinforcing this idea that the bioprocess is not in any way changing the phenotype, the biology, the behavior of these lines. And again, I've shown two examples here, which is for an mTOR inhibitor and for paclitaxel. But again, we tested a panel of 7 different chemotherapeutics and saw the same pattern across the board. So just to summarize, the first part of this presentation is really just to say that, you know, one of the key technologies that molecular devices has is this now ability to offer services to expand organoid lines. So we can basically take your organoid line and make it into a 3D ready product so that you've got access to your organoids when you want to use them. And as I say, we validated our bioprocess to ensure that it's comparable to manual culture. The next step obviously is that those organoids are now being put into workflows and being shown how you can use them in downstream assays. And for that, I'm going to hand over to my colleague Angeline Lim, who will take you through exactly how these organoids are used.
I really appreciate everyone in the audience here. So I'm going to try to, go over my slides as fast as possible in a reasonable manner. So I'll be around to answer questions if you guys have any more about the slides later. So just following up from what Vicky has been talking about, it's what do we do when we get the organoids? What are use cases? What are the proof of concept studies we can do? And she has highlighted a lot of advantages and benefits of using these preformed organoids. But one thing that I want to point out over here is that when you do a manual culture process, you spend a significant amount of time in the expansion phase. Depending on your line, it can go from weeks to maybe even months of just expanding your culture so that you can get enough for your screen. So with these preformed organoids, you just, from start to finish, you can get raw data, I would say in 10 days, depending on how long you're going to do your treatment or compound treatment for. And one of the very popular endpoint assay these days for organoids is with cell titer glow, ATP assay. But I want to also show you sort of a more phenotypic, morphological based assay that you can use to get viability measurements. And so here's an example of a 384-well plate that was seeded with the CRC colorectal cancer organoids from Celeste. And they're stained with viability dyes, and they were treated with all of these different compounds at different concentrations. And then if we were going to zoom in closer to look at these organoids, you can see just the variety or the diversity of phenotypes over here. And this is what I think we're trying to capture when we do sort of an image-based profiling experiment over here. And so I'll come back to this a little bit later, but just very quickly, just to show you how you can do viability assays. You would use very standard commercially available live dead stains for your organoids, and when you do an endpoint assay, and then you image it, and then for analysis purposes, you can do a bright field imaging like what I did over here, create segmentation mask, and then get all the measurements or features from the different channels, express it as a ratio, and then you can get a readout of viability. And in this case over here, the larger the ratio, the more dead organoids you have in your culture. And even just qualitatively looking at this panel of images, you can see that doxorubicin definitely is dead. Yeah. And then one of the advantages too of doing image-based analysis or image-based kind of endpoint assays that we can do monitoring over time. So in this case, you can get an idea of dynamics or the kinetics of what the compounds are doing to your organoids. So this is some examples, DMSO, 5-fluorouracil, and doxorubicin. So it's very clear that doxorubicin has very adverse effects on these cultures. They hardly grow over time, so obviously cytostatic effects. And let's see, next slide here. And because it's an image-based approach, we're able to also get object-level data. So every single organoid can be represented as a graph over here. It's, every point is a single organoid. And here you're looking at sort of the y-axis as representation of area of organoids over days in culture. So right in the middle is a DMSO control, and you can see over time they start to, of course, get bigger. Whereas those that have effects from the compounds they're treated with, they kind of just have a flat spread over time. There's no change in growth. And so I think, and this is sort of a summary graph of all the data we have over there. And so in short, you can see a lot of these compounds actually have significant cytostatic effects on organoid growth. And only one of them is doxorubicin, has a combination of both cytostatic and cytotoxic effects. And so I'm going to kind of switch gear a little bit more now and talk about analysis, because I want to introduce the idea of image-based, deep learning-based segmentation to help us identify organoids that's growing in culture over time and not having to be dependent on fluorescent images. So here are some example images that I think sort of highlight the difficulties when you come to image analysis of objects that capture a bright field microscopy, so non-fluorescent based. Obviously, there's very heterogeneous background. There's a lot of edge and well effects. And so it makes it really hard for someone to come up with a set of parameters that will help you identify the objects in an automated fashion. And so usually, in other words, a set of global parameters would rarely work for the experiment. You find yourself having to go back to the images over and over just to make little changes so that you can get a nice segmented images. And so this is what we think would be one of the better solutions for carrying out image analysis, and that's to use AI tools. So you can use deep learning-based segmentation to help you really robustly identify all the objects you want to measure. So I'm not an AI expert, but from a end user point of view, what you do is you load the images, step one, and step two, you annotate the images. In the software, you have these almost like Photoshop paintbrush tool where you would paint or color the objects you're interested in identifying. And next you put it through the deep learning neural network that's in the background in the software, and it does the training, and then you check the results. So obviously in 3 you can see that there are still some parts of the images where it picks out sort of that little cavity areas around the spheroids, so it's not great. You go back and do some retraining, and at some point you would get this perfect model, and you can save it and use it for further experiments or future experiments. Now, I get a lot of people asking me how many rounds of training do you need? How big does a training data set have to be? And I would say because our software comes with some pre-trained models, you work off these pre-trained models, and so sometimes as little as 5 to 8 images is enough to give you a pretty good model that you can use on future experiments. And so I'm going to show some examples of this deep learning-based segmentation tool that's in our Encarta software. So again, on the top panel, these were the colorectal cancer organoids that I showed earlier, but this is with 4x, so you can see it's kind of an ugly image. You have these shadows and shaded areas on the bottom right, just from the meniscus effect and just from being near the edge of the well, and there's debris there too. And here's a segmentation mask that we acquired from using one of the models that we built. Now, the bottom panels, I kind of like to show this because if you can see there in the orange box on the left of the screen, you can see these little dots that are picked up in the shadows. And initially, I was concerned because I thought it's picking up artifacts. But when I zoom in closer, change the contrast, I realized that it was actually picking out real organoids that I wouldn't have seen otherwise by eye. And then here's another example, which I hope you can see, is it's also picking out organoids that are trapped beneath your matrigel bubble. So typically, when you're creating these matrigel layers, bubbles are terrible. But over here, I was actually pleasantly surprised that it was able to still identify my object, and I can get readings from it or features from it. So for the example, on your left over there, that's human pancreatic organoids that were grown in these microcavity hydrogel-filled plates that are from Sun Biosciences. And so I like this because we could actually apply this to getting different results from it. So first we can get a model. We can use a model to identify all the microcavities, and that was done by Robert, who's sitting out there in the front. And then we can also identify individual organoids. And then if you look at these organoids, you would see that it's got some clear lumen or cystic areas. And we wondered if that was an interesting thing to track as well. And so Robert created another model that would identify these little cystic or lumen structures. And we did find that those organoids that were treated with toxic compounds eventually had fewer of these cystic structures. So it was actually a good readout and good thing to take note of in the experiment. Another thing we can use deep learning for is IPSC colonies. These are actually pretty hard to segment because these colonies tend to be very flat, have low contrast, and so you can create deep learning models that will help you identify them. Lung organoids, very similar. You can do the same thing. They're tough because there's a lot of background from out-of-focus organoids. You can do the same thing with mouse intestinal organoids, or just human intestinal organoids would work too. One thing to point out too is that I keep showing these bright field images, but it will work for fluorescent images too if that's what you want to do. I just find that it's very useful for bright field. So back to this, I showed this earlier and I talk about how they're so diverse phenotypically, and so how do we try to capture the phenotypic profiles, right, from the images. I'm just going to have one slide showing what we can do with these images in terms of data analytics. So with image analysis, you can capture and measure all of these different measurements. It's not a target-based kind of approach. You're just gathering all the information, gathering as much as you can. And then you will put it through data analytics platform where you can do something like feature selection, quality control, normalization, and data reduction, and get your principal components, hit selection and clustering. So for this, we actually partner with our collaborator as a company in Netherlands, Lab Analytics, and we use the cloud-based StratoMiner platform. And just sort of quickly, just the visualization part of it, the top scatter plot over there, in the purple bar, that's the DMSO negative control, and then compared to a positive control, that's the orange little blob at the top. So you can see that, your positive and negative control are actually very phenotypically distinct from each other. So that was really nice to see. And on the bottom of here, these are the U maps in a three-dimensional scatter plot. And you can see too, the orange dots are the positive controls. They cluster separately from the DMSO control. And then if you look here is a the doxorubicin treatment, and you can see sort of, the ones that are applied with low concentration are closer to the DMSO control, whereas the ones that were used that had high concentration of doxorubicin are closer to positive control. And on the right is just the same thing, but you're looking at a heat map of the concentrations that's being used. And of course, you can also do clustering with this approach, and this is really nice for when you have compounds with maybe unknown mechanism of actions, and if they cluster together very routinely all the time, it gives you an idea of what the mechanisms of actions are, and that can help you make more informed decisions later. And so Vicky has talked about the barriers to organoid adoption, and there are all of these reasons behind why it's not widely utilized these days. But I think at MOLEF, what we're trying to do is to deal with the technical problems, because we think we can just address some of these issues right now. And so I think one way to go is, of course, with automation. When you have automation, everything is scalable, is reproducible. It gives you so much control over all of these steps. So just a quick example about using automation is, this is just a very simple 2D cell painting assay that we use on a liquid handler. And if you just look at the hands-on time, if you're just doing this with a nice 6-well plate, it takes you 60 minutes to finish it. With automation, it was just 10 minutes hands-on time. Just put your plates there, put your reagents there, click start, and it does everything. And so because of that, about 2 years ago at Molecular Devices, we started out building our automation system, or our automation business. And right now we have at the San Jose site in the lab, what we call the Organoid Innovation Center. And this is what I get to work with, which is really nice. And I'm going to just show a quick video of the automation. Yeah, here you go. So it basically has a plate handler, which is this precise flex arm in the center. And then it can pick up plates, move it from the incubator to the liquid handler, trigger a process such as feeding. And then it can move that to the imager and trigger off an imaging protocol so that you can get images of whatever is growing. All right. And so this is something that we can do with organoid workflows. So mixing of organoids and matrigel all the way to the end point, it can be done in an automated fashion using that kind of setup. And so at this point, after 2 years, I'm actually really happy to say that we can automate most of the cell culture processes, from seeding to feeding or monitoring, and then to also passaging, because some lines has to be passaged, iPSC cells, or even intestinal organoids, we can do that. It did take a while to get there, but I'm happy to say that we have good results. Let's see if this plays. And the bottom panel, if it will play, this is the crash. What else can go wrong? Yeah, there was a video to show that the organoids grew over time. And let's see. There was someone trying to play videos yesterday, and it crashed all the time too, and she stopped playing videos. I'm just gonna go. All right, so what are we gonna do at the end of the day, and what's this? We're launching a new product, and I'm here to give sort of a sneak preview of that. And so while we can do all of that automation setup with these different parts, as a user, it will take you a significant amount of time to optimize and figure out what's the best way to implement it for your applications. So instead of that, we have taken all our learnings in the past 2 years, and we decided to package all of that into sort of a all-in-one product for you, and we call it the Cell Express AI, that's capable of seeding, maintaining, monitoring, and passaging your cultures. So let's see. So this is what the product looks like. It comes with a liquid handler in the instrument, so that's the one in the blue box, and it has an incubator in the red box. You can't see it, but it's in there. And then there's an imager there as well. So essentially, you will be able to do liquid handling, you can image, and also you can use AI-enabled on-the-fly analysis, like similar to what I showed earlier with the deep learning-based analysis. And then I've got some examples of the workflow we have implemented on the automation system. So one of them is iPSC culture and passaging. Here's a video of the iPSC feeding and passaging process. And so we have the plate tilted so you can really remove all the media from the 6-well plates. And here's another one. We've got customized reservoirs on deck, so you could actually put, I can't remember numbers, but you can put quite a lot of reservoirs on deck, and they're also temperature controlled. And after this is done, it goes back into the incubator that's below the liquid handling deck. And then here's the imaging with monitoring of iPSC cultures, and it's based on a deep learning-based segmentation that I showed earlier using Encarta, but it's embedded in the software. And then you can also have decision-based kind of rules set in there so that, for example, if your cultures have reached a certain % confluency, it will trigger an action. It can trigger passaging, or if you prefer to email you, we can do that too. Like, come to lab, passage your cells. So let's see. And then just the last part here, we will show some examples of 3D biology workflows. Here we're showing an example of seeding organoids in matrigel domes. And for those who have done it manually, you would know that working with matrigel is finicky, it's quite painful. And here we can get very defined domes in specific positions too in 24-well plate. We've done this with 96 and also 384. And then you can also continue to feed your culture by changing media, and this is an example of that. So with this, you have to move the tips to the side, remove the media, and then put fresh ones in. And let's see. Yeah, and we can also do intestinal organoid passaging. So this is one of the most complex protocols that we've implemented because you have to break down the matrigel dome, recover the intestinal organoids, fragment them, and then mix them in matrigel and reseed them again. And so this significant amount of optimization involved for this protocol, and I'm very happy that we got it. And example of some images, sort of on-the-fly images that can be acquired through monitoring and analysis, and there's a sort of nice readout where you can track the growth and what's going on in your cultures over time, so it's really great for QC purposes. All right, and so in summary, we believe that organoid technology holds a lot of promises in transforming drug discovery. There's still barriers to adoption, but at MoDev we're trying to address a lot of these barriers. So large-scale organoid production can be achieved. Talk to Vicky, and that will allow us to have standardized processes, standardized organoids that are uniform and nice to use in your screens. Of course, automation can be easily, or not easily, but can be effectively deployed across all steps in this screening workflow. And so just combining all of this together, we believe will be instrumental in driving research and drug discovery efforts. And here's the acknowledgement. We have great collaborators. Some of them are here. Thank you, Natalie. Core Life Analytics to help us with the data analytics. And of course, in MoDev, we have a large team just, applications team and also the hardware and software team in Salzburg. So, questions? Hi.
Yeah, hi. Thank you very much for the presentation. I think you showed nicely how, well, on one side you can scale the production, and then on the other side you also have like nice equipment and instruments and software to have a look at what's going on then during the essay. My question is about like the middle part, so during the essay itself, you also need a platform to do that. And you showed, I think, two examples now. One is micro well plates, and the other one just a well plate with a matrigel layer or a dome. And I wonder, what is your opinion about that? Is that, in your opinion, like, is there room for improvement in that section as well? Especially if you mass produce organoids, that you have a good culture vessel, that you have them maybe homogeneously distributed, maybe like quite in the same focal plane, maybe without matrigel, so that they can be easily be retrieved or accessed. So I just wonder what your opinion is.
So are you talking about the labware or the I mean, like, what kind of applications are you thinking of? Because I think with this setup, a lot of our endpoint assays is image-based, right? It's image-based. If you want to do more of a biochemical or even a plate reader-based, luminescence assay, it can be done too. What you do is hook up the plate reader into the automation system, use the liquid handler or plate washer to do the processing, and then feed the plate to the plate reader.
Yeah, so, and for, say you would want to retrieve the sample, is that something that you envision as well eventually to be able to have a workflow that allows you to get them out again?
Yeah, definitely. I mean, we do have some of that implemented already with the matrigel embedded organoids, iPSC2 by retrieving the cells. So those are retrieval for the next step, but we can also retrieve them for other purposes too, right? Yeah. Mm-hmm. No more questions? Hungry? All right then. Thank you so much for your time. Any questions, we're at the booth, so we can have more discussions if needed. All right.